Hi everybody. Today I'm going to talk about optogenetics. Optogenetics is a technique which combines optics and genetical engineering. The neurons can be genetically modified in order to be sensitive to light. Through this technique, you can control specific neurons of your brain just thanks to a LED light. So it is like switching off and switching on your brain. But how does it work? First, you must know what opsins are. Opsins are transmembrane photosensitive proteins. And firstly, researchers have looked for opsins in prokaryotes, for example in bacteria, like in halobacteria, and in algae, like in a member of the Chlamydomonas. And in the halobacterium, they have found NAPH, which is called halorhodopsin. And in the Chlamydomonas, they have found CHR2, which is called the channel-rhodopsin 2, because there is also a channel-rhodopsin 1, but it doesn't matter. And the halorhodopsin is an inhibitory opsin, while the channel-rhodopsin is an excitatory opsin. So let's see what they are. Inhibitory opsins. There you can see a membrane made of two layers of phospholipids, and inside and in the middle there is the opsin. To have a connection between two neurons, you need to have an action potential. In order to create an action potential, you need depolarization. Depolarization happens when the inside of the membrane is positive. But there you can see that this opsin, like halorhodopsin, is a cation pump, and so the ion chlorine goes inside inside the membrane when the opsin receives light, and so there cannot be an action potential. Here you can see an image of a neuron with the dendrites, the nucleus, the axon, and the terminals. And there you can see that there cannot be a connection, so there cannot be a synapse. Talking about excitatory opsin, you have the same membrane, and there you can see the sodium and the potassium. First look at the sodium. As it is an excitatory opsin, when it is hit by the light, the sodium goes inside, so the membrane is polarized, and there can be an action potential. After the sodium, the potassium goes outside, and then you have the hyperpolarization, which is the second phase of the action potential. But the important thing is that you can have the beginning, because the inside of the membrane gets positive because of the sodium. And here you can see the same neuron, and finally synapses can connect, so you can receive the message. Okay, let's make an example of how optogenetics could work. First, you have to take a gene, for example the gene that codificates for CHR2, and a viral vector. You put this gene into the virus. Common viruses are adeno-associated viruses and lentivirus. So here's an example of a lentivirus, which has got RNA genome because it is a retrovirus. And in this way you can have a new virus because you have a new RNA with the CHR2. And so this virus infecting a cell will promote the expression of this protein. You infect a mouse with this virus. The virus must be injected into a particular area of the brain, for example, the center for hunger, for movement, particular feelings, because you want to infect only those neurons. And also the gene that codificates for the opsin must have also a promoter, because the protein must be expressed only in the cells that we want. And then you have this lovely mouse and you take a LED light that must be of the right wavelength, because each opsin opens at a different wavelength. Channel rhodopsin opens at with the blue light at 450 nanometers, but allorhodopsin open at with a green-yellow light. And you put inside of the brain of the mice fiber optic cable and an electrode, and then finally you can switch on the light. What could it happen? We put the gene for CHR2, which is an excitatory opsin, so synapses can connect when the channels are open, and so the mouse is hungry. But if you do not have light, the mouse is not hungry. How could optogenetics affect our lives? First, we have opsins. We have opsins in our eyes, in the retina, because in the retina we have cones and rods, and opsins can be found in rods, and they receive the light and send the message to the brain. This is the reason why optogenetics at the beginning has been applied mainly in retinal pathologies. But it is also an extremely useful tool to understand the mechanisms of our brain, because if you can switch on and switch off different areas of the brain, you can understand what they are important for. And you can also find therapies for memory loss, which is amnesia, or drug addiction, Parkinson's disease. Nowadays, optogenetics is too invasive to be used inside human brains, because you should put a fiber optic cable right inside of the bone, and so it is quite dangerous. But tomorrow it won't. It is a great challenge for the future, because optogenetics has got a great specificity. You can switch on particular neurons, not just make a deep brain stimulation like it is nowadays. But this techniques makes me hope for the future, because through good tools you can have outstanding progresses in medicine. And so I found it really exciting. I hope you liked it and enjoy.